Bismillahirrahmanirrahim. Assalamu alaikum everyone. Today the topic of our discussion will be type of blood groups, and the reason of selection of this topic is because this topic is very easy to understand but seems complicated when we start reading about the blood types. So I will try my level best to clear it for you guys. So the basic types, the most common types like ABO blood group system, RH factors, and the Bomby phenotype, which is a rare type. Bomby phenotype and then Parabomby phenotype. I will try my level best to clear this type of topic for you guys and try to make it easy for the students as well for their understanding. The basic introduction is that if you talk about the ABO blood group system, your blood group depend upon the type of antigen found in your RBCs. Like if the antigen A is found in your RBCs, then the blood group will be A. And if the antigen B is found on your RBCs, the blood group will be B. And if both of these antigens are found on the RBCs, then the blood group will be AB. And if both of these antigen, like antigen A and antigen B, are not found on your RBCs, if both of these are absent, then the blood group will be O. This is about the ABO blood system. Now, oftenly we talk about the blood group is either my blood group is A positive, and someone else blood group A negative, AB positive, O negative, O positive. So the positive and negative is not actually having any concern with the charge, because the student often interpreting this positive and negative as a charge. No, this is not charge. The positive means the presence and the negative means the absence. And what type of antigen? The RH antigen. You know, rhesus monkeys first time discovered in the blood of rhesus monkeys by the person called Landsteiner. So if along with these antigens like ABO, if the RH antigen is found on your RBCs, then the blood group will be positive. And if the RH antigen is absent, is not found on your RBCs, then the blood group will be negative. Like one person having antigen A on the RBCs plus RH antigen on the RBCs, the blood group will be A positive. If another person have B antigen along with the RH antigen on the RBCs, the blood group will be B positive. But if have only A antigen but no RH antigen, the blood group will be A negative. Only B antigen but no RH antigen, the blood group will be B negative. Similarly, AB antigen along with the RH antigen, the blood group will be AB positive. And if only AB antigen but not RH antigen, the blood group will be B negative. Similarly, only RH antigen plus the H substances, I'll explain the H substances, then the blood group will be O positive. And if only the H substances but no RH antigen, the blood group will be O negative. Since the positive and negative is actually representing the presence and absence of RH antigen on the surface of RBCs. Now, in the next video, very next, I will explain what are these antigens and how they get assembled. So viewers, this is what we were talking about. The person blood group is actually depend upon the type of antigen found on the RBCs. Just take over here. Look, when a person have only antigen A on the RBCs, the blood group will be A negative. So another person having antigen B on the RBCs, the person will have B negative blood. Another person have both of these antigen like antigen A and B will have the blood group AB negative, while the one who do not have these antigen like antigen A and antigen B also do not have the RH antigen will have the blood group O negative. So A negative, B negative, AB negative, while the O negative, the A, B and AB actually depend upon the antigen A and B, presence or absence of these antigens, while the negative actually depend upon the RH antigen. As you can easily look at these RBCs, that RH antigen is not found on these RBCs, while on the other hand, if we talk about this section, look at this, the section have both RBCs have both antigen A as well as RH antigen. So presence of RH antigen indicating the blood group is A positive. So A because of antigen A and positive because of RH antigen, and same in the case of B as well, the B antigen and RH antigen, the blood will be positive. Both of these antigen AB and RH antigen AB positive, while only the RH antigen is found on the RBCs, the blood group is O positive. So this is what about the positive and negative. Now the next we will discuss about how these antigen get assembled, especially the antigen A and antigen B, because our main focus is about these antigens like antigen A and antigen B. Okay, so this time you need to follow everything what I'm explaining in front of you. Look at this. This is one of the RBC, red blood cell, and you know, in their immature stage, these cells do have nucleus. So inside the nucleus there are chromosomes, and chromosomes carry gene. Those actually transcribe in the language of protein, and those protein will assemble in the either enzymes or regulatory proteins. So this is one of the RBC, and this RBC, red blood cell, have chain of oligosaccharide, whatever shown in the green. This is chain of oligosaccharide. Look at this: glucose, D-galactose, N-acetyl galactose, and this galactose, oligosaccharide, chain of carbohydrate. Remember, this chain will be present on your RBCs. Red blood cells, they do have this chain of carbohydrates, oligosaccharide, chain of oligosaccharide. Now this chain of oligosaccharide will be modified into the antigens, either in the antigen A or antigen B. This will be modified, but this is the basic structure that you need to memorize, that RBCs do have this chain of oligosaccharide, glucose, D-galactose, N-acetyl galactose, and this one galactose. How these oligosaccharides will be modified, we are going to explain it, so be ready, attentive. Okay, for the modification of oligosaccharides, multiple genes are involved. Say like 2 genes are involved. One of the gene, the H gene. This H gene is found on the chromosome number 19. And you know, when this H gene will transcribe, it will produce enzyme. Which enzyme? Fucose transferase. Fucose transferase. As the name indicating, the transferase enzyme will transfer fucose. It will transfer this fucose into the oligosaccharide. Transfer this fucose into the oligosaccharide, and after getting this fucose, now the chain of oligosaccharide which is found on the RBCs, now this chain will called as H antigen. H antigen. How the oligosaccharide will convert into H antigen? When the fucose will be added into this chain is now called as H antigen, and that's it. H antigen. H antigen. Further modification is actually performed by the product of another gene, and that gene is found on the chromosome number 9. Look. Okay, as I stated, now we have H antigen on the surface of RBCs, and the further modification of H antigen will be performed by the product of this gene, the I gene. This I gene is found on the chromosome number 9. And here I took the 2 major alleles of I gene, like allele A and allele B. One we inherit from mother side and other we inherit from our father side. Okay. Now, this allele of gene I will transcribe into enzyme N-acetyl glucosamine transferase, which is also called transferase A. N-acetyl glucosamine transferase A. And this allele of I gene will transcribe into enzyme galactose transferase, or you can say transferase B. Transferase B will have 4 less amino acid as compared to transferase A. That is the structural difference, but we are not going to. Look at this, both of these are transferase. Just check transferase A and then transferase B. But what about this prefix? N-acetyl glucosamine. Mean transferase A will transfer the N-acetyl glucosamine, while transferase B will transfer the galactose. Where they will transfer? Yes, they will transfer these things into the H antigen. They will transfer these things into the. Okay. N-acetyl glucosamine transferase A will transfer N-acetyl galactose into this guy, mean H antigen. After having this, the H antigen will convert into antigen A. Antigen A, how the antigen A will form? Chain of oligosaccharide plus fucose, chain of oligosaccharide plus fucose will make the H antigen. H antigen plus N-acetyl galactose. H antigen plus N-acetyl galactose will equal to antigen A. This will assemble into antigen A. Well, what about the antigen B? Very simple. Transferase B will add this galactose into the H antigen, and after having this galactose, the H antigen is not H antigen, but now it is B antigen. B antigen. Okay, to simplify it more, B antigen is equal to chain of oligosaccharide plus fucose. Chain of oligosaccharide plus fucose is equal to H antigen. H antigen plus galactose is equal to antigen B. So now this person will have both of these antigen, like antigen A and antigen B. When one of the person will have both of these antigen, the person blood group will be AB blood group. So this is how the AB blood group get assembled. The rest is very easy to understand. How the rest is easy to understand? Look, if the person is having these alleles, one from father and other from mother, and they are homozygous state, AA, definitely this form of I gene will synthesize only N-acetyl glucosamine, mean transferase A. When the transferase A is there, the transferase A will transfer N-acetyl galactose into the H antigen. Then H antigen will convert into the antigen A. So the person will have only antigen A on the RBCs, the blood group will be A. Same in the case of B. If the person is actually having both of these alleles, one from the father side and other from the mother side, the same type of allele in their homozygous state or in the heterozygous state, this form will actually is non-active, you can say recessive form. So this person will transcribe only the transferase B, galactose transferase. So when it is transcribing, it is synthesizing the transferase B only, the transferase B will transfer the galactose into the H antigen, and after having galactose, the H antigen will convert into the B antigen. So the person blood will have only and only B antigens. So blood group will be B. And the third one I already explained. And this one, O antigen, how the O antigen will produce? The person is not producing either transferase A or transferase B. and the person blood will have only H antigen. How the H antigen? Look guys, H antigen means that chain of oligosaccharide plus fucose. But further modification of H antigen is not taking place. Why? Because the further modification of H antigen require the second enzyme, either transferase A or transferase B. So no more modification in the H antigen. The person blood will have only the H substances and the blood group will be O. This is how the ABO blood group get assembled. Now the next we will talk about the Bomby phenotype. How the Bomby phenotype can happen. The Bomby phenotype. You know, Bombay because of Mumbai, one of the Indian city. In 1952, one of the doctor, I think Bende, observed certain people have such type of the phenotype that genetically or with respect to the I gene they are AB, but the phenotype, phenotypically they are O. How this actually is a rare type, very rare type. 4 out of 1 million people. How this happen? How the person can be AB with respect to the I gene, but O phenotypically? The RBC do not have antigen A and B. Look, this is because of recessiveness of H gene. This is because of recessiveness of H gene. How this happen? When the H gene is in its dominant form, either in the homozygous condition or in the heterozygous condition, this H gene will produce the fucose transferase. And the fucose transferase will transfer the fucose into the oligosaccharide, and after having this fucose, the oligosaccharide will convert into H antigen, and H antigen will be the substrate for those enzymes like transferase A and transferase B. Remember. But if the person is homozygous recessive with respect to H antigen, this person will not produce fucose transferase. When this person is not producing fucose transferase, the fucose will not be added into the oligosaccharide chain of carbohydrates or oligosaccharide. So this chain will not convert into the H antigen. So when there is no H antigen, both of these transferases like transferase A and transferase B will not able to convert antigen, to synthesize antigen A or antigen B. Look, transferase A and transferase B can add either N-acetyl galactose or galactose into the H antigen. And as they are not getting H antigen, they will remain functionless. They will not able to convert H antigen because there is no H antigen into the antigen A and B. So they will not be able to synthesize either antigen A or antigen B. Then the person blood will have, RBC will have only the chain of oligosaccharides, but no H antigen, but no antigen A and B. So with respect to the I gene, the person will be AB, but phenotypically the RBCs will not have antigen A or B. Phenotypically, such a phenotype is called Bomby phenotype. Genotypically AB, but phenotypically O. How it happened? Because both of these transferase A and transferase B, they are not getting their substrate for the addition of these things like N-acetyl galactose or galactose. RBCs have only chain of oligosaccharides. Why the oligosaccharides are not converting into the H substances? Because of recessiveness of H gene. And H gene synthesizes fucose transferase. Fucose transferase is absent. Fucose is not getting added into the oligosaccharide. Oligosaccharides are not getting converted into H antigen. So when when there is no H antigen, then the transferase A and transferase B will not be able to convert this H antigen into the A antigen or B antigen. So phenotypically the person will be O, but with respect to the I gene, genotypically the person will be either A or B or AB. Such a phenotype is called Bomby phenotype.